drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. The full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode.
You start the podcast? It was within the first 10 episodes. You had Lisa Montegio was one for sure.
Yeah.
And there was maybe Carl Harris. I can't remember. There's a couple others that, I was first in pretty early stages of our research at WashU and listened to those, and they were like really useful. I'm very much of a auditory learner, so I love podcasts.
Okay,
I've
great.
read some of these people's papers, but I think hearing somebody talk about their research, it brings it alive in a very different way, so listening to those really, help me conceptualize how these great thinkers in the field are thinking about some of these questions.
Yeah. And what's nice about talking to someone too is, as opposed to reading an individual experimental paper or even a review paper, they bring to the whole thing all of the contextual and historical knowledge they have. So it's a lot easier to understand particular findings and particular observations with all of that context brought to bear.
Yeah, absolutely.
So
more engaging, and it's probably more useful.
Yeah.
Brian Roth. I was just checking. Brian Roth was another one.
Yeah, that was an early one I had. So I essentially started at the very beginning of 2021. And so those were among the first ones. Lisa, with probably the first episode I did, focused on ketamine. And then Brian Roth was a very early episode, I think within the first 10.
Yeah. We were I had just started, I was in psychiatry residency. And we had written some small grants, like pilot grants, to do psilocybin, human clinical research at WashU. So we're and then basically, depending on when it was, we got the grants, and then everything got, frozen with COVID. So then I was just had some time to really immerse myself in the field.
I see. How did you sort of get into the field? What was prompting that, or what were you doing before?
Well, my background is in systems neuroscience and basically fMRI, and WashU. They have a particularly good, program in resting state fMRI and fMRI cognitive neuroscience, and so I had helped build some of the tools for what we call precision functional mapping. And I was really interested in psychopharmacology, and that was like the big picture plan was I want to take these next generation human fMRI approaches and use them to understand drugs better. And it seemed like I was going into psychiatry, and ketamine and psychedelics seemed like the most exciting direction to go with that. So
yeah,
I saw some of the Carhart Harris initial papers, and they were exciting and tantalizing, but I was like, we could take this, I think, quite a bit deeper.
Yeah, yeah. I want to get into maybe we can start with some methodological stuff to build a base for people to understand some of the recent results you've gotten around psychedelics. So, fMRI, functional magnetic resonance imaging. I want to talk about the sort of the specific precision mapping stuff that you just mentioned, but let's just start very basic here. Can you talk a little bit about the basics for how fMRI works and what exactly it's measuring and not measuring?
Sure. Is this official? Are we
Oh yeah,
recording?
we're going.
Okay, all right. So it's a indirect measure of neural activity, right? So it's measuring changes in blood flow. And this is the most important caveat to understand about it. And so anything that changes blood flow changes fMRI signal. But the fundamental, assumption is that if a part of the brain becomes more active, there is a very well-oiled machinery that increases blood flow. I mean, down to the level of, a millimeter, basically, for that population of neurons. Um, and the actual increases in oxygenation of blood. So there's this big increase of flow of oxygenated blood, right? And so what fMRI is actually measuring is blood oxygenation, which goes up when there's neural activity. And that's like the first basic thing to understand about fMRI. But then the next basic thing to understand is most of what I do and most of what we're talking about is this idea of functional connectivity. And this is the idea that if you just look at, let's say, two parts of the brain, let's say one on the left hemisphere, we'll take the visual cortex, right? Left visual cortex and right visual cortex. And you look at the kind of resting fluctuations in this fMRI signal. And what you see is that if those parts of the brain are communicating and are basically co-activating, you have a very correlated, you know, up and down fluctuation of this fMRI signal. And that's the two area example, but you can scale that up all the way to the level of the entire brain and come up with these pretty intricate maps of brain networks, So you scale that up and you get the visual system, scale it up further, and you have attention systems, motor systems, and then these kind of executive function and default mode network has been one of the big sort of revelations that's come out of this resting state fMRI approach. And so it's a powerful systems neuroscience tool for understanding, the large scale brain in humans, what's going on when you're, in under different disease conditions, what's going on under different drug conditions.
Mm-hmm. So fMRI is measuring, it's not measuring neural activity per se, but the idea is, neurons become more or less active in response to what an organism is doing. So if a neuron starts firing more, this is a very energetically expensive process. It requires a lot of ATP. And therefore, when a chunk of tissue becomes more active in the brain, blood flow has to increase to that chunk of tissue in order to reinvigorate the tissue with oxygen and nutrients and all the stuff that's going to sustain that very metabolically expensive process, the information transmission mechanisms of what neurons do. But what we're actually measuring with fMRI is not directly the neural activity, it's the change in blood flow to different areas, which we assume or know to some extent or another is going to correlate highly with neural activity.
That's right. Yeah, yeah, that's right. If you really want to get into the weeds, which you probably don't, but there's a magnetic pulse that aligns the electrons and the rate at which deoxygenated blood, the electrons in deoxygenated blood relaxes versus oxygenated blood relaxes differs, and that's what kind of creates the fundamental contrast of fMRI.
Mm-hmm. Are there any ways that the fMRI signal can start to mislead us if we rely too heavily on the assumption of a one-to-one correlation between blood flow and neural activity? So, for example, are there any known examples where you can have more or less purely or largely vascular changes that are actually not so well correlated with underlying neural activity?
Yes, there absolutely are. And this was
Mm.
one of the fundamental things that when I started doing psychedelics research, one of the fundamental questions was these studies have been coming out showing, some cool changes in functional connectivity, let's say, some cool changes in, evoked brain activity with fMRI. And but at the same time, if you dig into the neurobiology of the 5-HT2A receptor, this is a receptor that's actually expressed on vasculature as well. And we know that
Mm.
like, at high doses, psychedelics can be vasoconstrictive, and they can change, basal blood flow. They can also maybe change the relationship. What's less known is whether they change the relationship between neural activity and blood flow. Um, but that was for sure one of the big questions. And then actually that's been a side project. We have a paper that's under review at Nature Neuroscience right now, which gives a very compelling answer to this by simultaneously measuring fMRI and neural activity.
Interesting. So the so-called psychedelic receptor, the serotonin 2A receptor, which is very important for the effects of serotonergic psychedelics. This receptor is not only located on neurons, it's also located on blood vessels themselves. And so a psychedelic could bind to this receptor on blood vessels and induce changes that you would pick up in an fMRI signal that may not have anything to do with the neural activity in that pixel of the fMRI image.
Yeah, that's exactly right. I mean, it's expressed in a lot of places. So
Mm.
astrocytes, which they are indirectly related to, vascular response. It's also have now somewhat lower than the level that the 5-HT2A receptor is expressed in kind of pyramidal neurons, which are the primary excitatory neurons in the cortex, but it is expressed in astrocytes, and it has all these different functions. And, like a fun thing to keep in mind is that serotonin, so it's a serotonin receptor, right? And the name serotonin is actually a combination of serum and tone, because the way it was first discovered is the fact that it exists in blood and it increases the tone of blood vessels. So in other words, it
Mm.
causes blood vessels to constrict. So this is like, this is critical information if you're trying to study what psychedelics do, particularly if you're trying to use changes in blood flow and infer that those are representing changes in neural activity.
Hmm. And so, how do you understand and parse those two things? Are the studies that you're referring to the type of work that you mentioned? Is it animal work where you can do both fMRI imaging and direct measurements of neural activity using electrodes or things like that?
Yeah, well, we have one. So I worked with a group, Adam Bowers, the PI, but they have, animal imaging facility, and they can do simultaneous, uh, you know, calcium channel imaging where you get basically fluorescence anytime a pyramidal cell depolarizes, and blood flow imaging at the same time. So you can get both the neural activity and the blood flow, and actually map out the relationship between the neural activity and the blood flow, which, like we said, under typical conditions is very consistent and stable. And so we did some studies assessing if and how that changes with a psychedelic,
I
partly
see.
because that's like step one. If you're going to use fMRI to try to make assertions about how psychedelics are changing brain networks or brain connectivity, you have to know that, are you looking at changes in blood flow, or are you looking at changes in neural activity?
Hmm. Hmm. And so, when you do functional mapping using fMRI, you're using this blood flow signal that's a proxy for neural activity. And when a human is engaged in something, a visual task or whatever, this will induce different chunks of tissue in the brain to become more or less active. And there'll be like a systematic pattern to this. And so you can sort of infer, the idea is that you can infer patterns of literal, like anatomical connectivity, essentially, from the correlated change in signal in different parts of the brain.
Yeah, that's right. So, the fundamental idea of functional connectivity is that you can look at resting fluctuations in activity, and from that you can infer patterns of, you can infer brain networks, you can infer patterns of connectivity, But then the next step for that is that, if you want to understand how a drug is changing brain networks, you can look at how those patterns change in response to the drug and infer how that drug is altering. And of course, that's the big question with psychedelics, or a big question is what is causing this very unique subjective experience that psychedelics produce.
Mhm. And one of the things that you read a lot about in the literature, the fMRI literature, is um, networks that arise under situations where there's task engagement versus no task engagement. So, for example, obviously, if you engage someone in a visual task, they have to look at things and use their eyeballs and their visual system. The visual system is going to sort of light up and be engaged in ways that it wouldn't otherwise be if you instead give them a task that's an auditory task. Obviously, the visual and the auditory systems are going to have different levels of activity reflecting the use of the auditory channel versus the visual channel and so forth. But even if you give someone nothing to do and they're just sitting in the scanner at baseline, there's still patterns of activity there that reflect this non-engagement in a task. And that's where you start to think about things like the so-called default mode network. So what exactly is the default mode network? How is it discovered? And what is it really?
Well, yeah, so I'm going to start by building off of the first thing that you were describing, because I think it all fits together, right? Which is this idea that even, free of like a specific task or a specific, let's say, function, the brain is always active, right? And for example, when somebody is at rest and not doing anything externally versus the most demanding cognitive task is maybe like a 1% difference in actual metabolic and hemodynamic activity in the brain, right?
Hmm.
So the brain's always active, okay? And the fundamental like way that the brain is active is that you have fluctuations in, you have collections of neurons that form kind of a big circuit, and those neurons are always working together and communicating together, and they are fluctuating, in terms of their activity, and they're kind of fluctuating between active local states and deactivated local states. And so that's the kind of fundamental unit is a collection of, we'll say, 100,000 neurons that are typically very synchronized and co-activating. And then you take multiple units or multiple brain areas, and they're also, if they are working together normally, for example, the visual system or the motor system, they're going to activate and deactivate in synchrony. And so this kind of understanding and the ability that you can measure this with fMRI to a pretty reasonable degree is what has led to us to be able to do kind of a full parcellation of the whole brain and say what networks are there, what are they doing. And one of the things that's emerged from this, which has been really influential in our thinking about what the brain does and how it works, is the default mode network. And this idea that there's a collection of brain areas. Um, we will stay away from the neuroanatomy. I'm happy to go into it, but we'll just say across both hemispheres and across, from the more anterior and more posterior parts of the brain, collection of areas that uh, are more active by default when you're not engaged in any sort of external attention demanding task, which is why it's called the default mode network. And this collection of areas, and then the way it was first discovered is that when you had somebody get in an MRI scanner and do whatever, task the experimenter told them to do, this collection of brain areas actually deactivated and decreased in their activity. But what we've now learned is that what these brain areas are doing seems to be critical to creating your sense of self. Okay, so these brain areas are like defining, who you are and allowing you to think about yourself in relation to your environment. So, for example, if you give somebody a list of words and ask them which words describe you, okay, when they are picking out words that describe themselves, the default mode network is actually more active. So, this has been why has this been a fundamental discovery? Partly because it looks like many psychiatric illnesses and probably some, neurodegenerative illnesses really fundamentally are illnesses of the default mode network, which fits into a lot of more psychological theories about kind of the role of self and pathology of the sense of self in depression and in other psychiatric illnesses, schizophrenia.
Mm-hmm.
So
What would be a sort of an example there? So, something that comes to mind for me is when you look at clinical descriptions of what major depression is, things that, it's often characterized by things like rumination. People are constantly thinking about themselves and what's wrong with them. Is that kind of the idea here, that the system is involved in yourself, your sense of self, thinking about yourself, attributes that apply to you, and if it becomes too overactive or active in certain ways, this results in pathologies where you're, say, negatively ruminating about yourself, and we call that depression.
yeah, that's exactly right, Nick. And there's been studies showing that in people with depression who are, ruminators, they have more active default mode network, and they have a more difficult time turning off their default mode network in order to engage in an external task. So that, yeah, it's exactly hit the nails on the head.
Yeah, and I would imagine too, like on an intuitive level, I think everyone basically knows or can know through introspection that if you're just sitting around, it's really easy to think about yourself and your life and this and that. But the more you're engaged in a task, the harder it is to retain that narrative around you and yourself. If you're playing a championship basketball game, you're not really thinking about yourself. You're thinking of, you're just engaged in the sort of sensory motor activity of trying to get that ball into the hoop.
Yeah, I found that to be interesting to think about. Like when exactly. So, you talk about the flow state. What is the flow state? In a large part, the flow state is like the quieting of the sense of self, right? And so that attention can be completely focused on engaging with your environment. And by contrast, turns out that the majority of time that somebody is mind wandering or daydreaming, the contents of that is deeply related to sense of self, either in the present, in the past, or in the future, right? And so that's why just you let somebody let somebody daydream and their mind's wandering, the default mode network is probably going to be more active.
Hmm. And so let's walk people through what what was sort of known in the literature leading up to some of the work that you've recently done with respect to how psychedelics affect the brain in general, in terms of what we know from functional imaging, but the default mode network in particular. What's the field? What are the results in the field that we think are relatively solid there?
Well, yeah. So, there was the idea that psychedelics are in particular targeting the default mode network. And, Robin Carhart Harris, who you've had on maybe a couple times as a guest, he both had some empirical evidence for this with some of the first fMRI studies that he did, now going back 10 years at Imperial College. Um, and also built it out as a psychological theory. And this comes to some of these, the entropic brain hypothesis and some of the, writing that he's done, um, in terms of putting together what is the idea that psychedelics are altering the default mode network, and, should we be thinking about how these drugs working. I think at an intuitive level, it makes sense that, they're disrupting a sense of self, and, people report obviously ego dissolution, and that's a core part of the experience. And so connecting that with the default mode network, I think, he laid some of the groundwork for some of those ideas.
So, and is there any worry with that pre-existing literature? So if we riff on what you were saying earlier about fMRI being, it's a blood flow signal that we're measuring. Blood flow is going to correlate quite tightly with neural activity most of the time, but not always completely. And you do have this additional potential problem here with the psychedelics in particular, which is that the vascular system, the blood vessels in the brain itself, are sensitive to psychedelics because they express the 5-HT2A receptor. Is there any worry that some of the results from the pre-existing literature might have some confounds there?
That was certainly a concern. That was certainly a concern. And, we did a number of things to test out that idea, including some things that we did in the human study, and also with this particular animal setup where we're simultaneously measuring neural and blood flow fluctuations. And, you know, I think there's two sides to this. One is that there are consistent changes to the impulse response function between neural activity and blood flow on a psychedelic, but they are relatively small. If you look at the sort of shape of the impulse response function, actually, it's not even what I expected. It's weird. It's a little quicker between after taking a psychedelic, which I don't even know what that means or how to necessarily think about it. But it is surprisingly consistent between mice and humans. So you have a slightly faster blood flow response to a change in neural activity. I mean, it's relatively, like I said, not a huge. It's maybe a few percent faster, and also more transient. So you don't get this longer lasting increase of blood flow. So there's a little shift, and it may account for part of the changes that we saw in humans. But one of the things that we did in the human study, which I'm jumping ahead a little bit. I can tell you more about how the study was laid out. But we actually had people doing some tasks while they were on psychedelics, so we could specifically, show, look, you still get a blood flow response to neural activity. It looks relatively similar to off of drug, and it looks basically like we would expect, and we can actually account for some of the changes to blood flow and still see that there's pretty dramatic shifts in functional connectivity and in the signal in general.
Mm-hmm.
That
What
makes
are
sense.
Yeah, that makes sense. So before diving into some of the work, the specific work here, I think it's worth mentioning, walking people through a little bit when we talk about the default mode network. What are some of the major hubs in that network? We don't necessarily need to go through all of them in complete detail, but what are some of the major hubs, and how do we normally think about the function of these different hubs of the network?
Sure. I mean, one of the big ones that's really been of interest in psychiatry and in emotion is the medial prefrontal cortex. And so this is a part of the brain that seems to be a critical link, for one thing, between the limbic system, so the hippocampus, amygdala, which are these sort of emotional and memory hubs of the brain, and the cortex, which is the higher cognitive thinking. And so there's a lot of sort of both theories and empirical evidence about how the cortex relates to these emotional structures, but a lot of that connection is through the ventromedial prefrontal cortex. So that's a hub of the default mode network, and it seems to be intimately involved in reacting to emotion, seems and processing emotion, seems to be intimately involved in empathizing with the emotion of others, for example. And then other parts of the brain, posterior cingulate and some parts of the angular gyrus, which are also have pretty strong links to, or pretty strong associations with, interpreting theory of mind, so thinking about what someone else is thinking about or understanding someone else's actions in relation to your own.
And so you published a paper recently in Nature looking at the effects of psilocybin on the human brain using fMRI. Can you set this up for us, both sort of conceptually in terms of the types of questions that were motivating this work, and then methodologically how you went about it?
Sure. So this was uh, started about, the kind of conceptualization for this study goes back around 5, 6 years now. And at that time, the idea of precision functional mapping was really emerging from the imaging group that I had been collaborating with at WashU. And I think that honestly, that's really the central critical advance to the work. And that's, to put it simply, this is the idea that the way most fMRI studies are done, you're averaging across a group of people, let's say in condition A, and looking at, how does the brain respond to some variable in condition A, and then how does the brain respond to some variable in condition B. So you take everybody, you take the data from maybe everybody in the first group who's exposed to condition A, and you average it together. And you take the data from everybody in the second group who's exposed to condition B, and you average it together. And the fundamental problem here is that everyone's brain is different. And it might sound silly almost, but even, the best possible technology and techniques that we have for registering brains together to a common space are still far from adequate. And
Mm.
the particular shape of the area in your brain that responds, let's say, to a face, right? You have this well-defined fusiform face area, but it's not going to be well aligned to the part of the brain that responds to a face in my brain.
Mm. So you want to
Uh,
align things functionally, but there's not going to be a one-to-one anatomical correspondence in terms of where these functional parts of the brain are in one brain versus another.
exactly right. Yeah. And so precision functional mapping is the idea of essentially doing all of your neuroscience and all of your analysis within the single individual, and only then kind of after you've looked at the effect of intervention, comparing, is it consistent between individuals? And so that includes getting much more data on a single subject and following them kind of longitudinally and mapping out, their connectome fingerprint and their brain areas, in my example, mapping out their fusiform face area, right? And then saying, what is the effect of my intervention within this person? And then comparing to your fusiform face area, is the effect of the intervention the same for you? Does that make sense?
Yeah, so I would imagine, say if I'm reading an old paper on psychedelics and the default mode network, what you have to do for each individual that's going into that study is you have to map out functionally where exactly their default mode network is, where the boundaries and links between all these hubs are. And you have to measure the effect of something like a drug, like a psychedelic, on their individual default mode network. And then you have to do it for all of the other individuals, but you have to do the mapping separately and specific to them because everyone's going to be a little bit different in terms of where that network is and what it looks like. And then you aggregate the results together and give average depictions or visualizations of what's happening. And that's typically what we're looking at in a paper.
Yeah, that's right. And the way, I mean, most of the psychedelic studies and most studies in general that have been done, you average brains together first, and what you see is, let's say, the effect of a psychedelic is this very fuzzy, blurry change in, default mode connectivity, or maybe fuzzy, blurry change in connectivity and association cortex. But by actually mapping these systems within an individual with a high precision, you can get much more detailed answers about which particular subcircuits. I mean, it's kind of amazing how just by sort of flipping this idea on its head and looking at things within a single subject, we've been able to define very nuanced and reliable subcortical networks in the thalamus and the basal ganglia and the cerebellum, and just has made fMRI a lot more powerful.
Mm-hmm. So you're able to detect things that would otherwise basically kind of get smeared away in the process of
Exactly.
analyzing this data.
Yeah, exactly. So, the fundamental question was, I think it was two things actually. One is, can we get a more detailed picture of what FM of what not fMRI, what psilocybin is doing that's producing the very unique subjective experience, what it's doing in the brain, in other words, that's producing the unique subjective psychological experience. And then the second was, what is the lasting effects after the drug has worn off? And, by getting these much more high resolution images, can we actually see changes that are lasting in the weeks afterwards? And part of the story there is that there's been all this fascinating data showing, we were talking about Lisa Monteggi earlier, and her and some others have shown that there's these lasting plasticity effects with ketamine and with psychedelics, which last for days, weeks afterwards. So the question is, what's that doing at the level of brain networks and kind of systems neuroscience?
Interesting. Okay, begin to walk us through the recent paper. So you did this precision mapping. You looked at psilocybin. What were some of the what were some of the questions you were asking that were new here that was building on the previous literature?
Well, yeah. So one was, what is changing in the brain when you take a high dose of psilocybin, both immediately and in the weeks after.
And what's a high dose here?
So it's 25 milligrams, but this is 25 milligrams of synthetic psilocybin, which, as I understand it, is probably close to 5 milligrams of psilocybin mushroom, dried psilocybin mushrooms.
5 milligrams or grams?
I'm sorry, did I say milligrams? Yeah,
Yeah, 5 grams. So that's big.
Yeah, and that's the dose. I mean, that's been the dose that I think all of the companies that are in later phase clinical trials are using as their kind of full therapeutic dose.
Yeah, Terence McKenna famously said that the heroic dose was 5 dried grams in silent
Exactly.
darkness, and yeah, I can tell you that 5 grams. I don't think I've ever taken 5 grams. 5 grams, that's a lot.
Yeah, yeah. So for one thing, it's miraculous that folks were able to tolerate being in a scanner for 2 hours
Hmm.
while they were on 5 grams of mushrooms.
Were they previously experienced with psilocybin?
Yeah, we decided to make it a criteria that they have to have had a psychedelic and, have had a relatively good experience at some time in the past. But we kind of walked a tightrope because we also said we don't want them to have taken a psychedelic within the last year.
Mm, okay.
So they had all had an experience with a psychedelic.
Okay. So what are some of the initial results here? So I believe for this study you did many scans per individual, and only a couple scans or so are with psilocybin. You did a lot of baseline measurements as well, I think.
Yeah. And by the way, so we did a lot of baseline measurements, which partly was so that we could get this very detailed connectome, individual connectome fingerprint for each subject. But one of the unintended consequences of that was, I think it actually made it a lot easier for people to tolerate being on this hero dose while they were in a, loud
Yeah,
bang scanner.
it wasn't the first or second time in there.
Exactly. So it was kind of a familiar environment. So actually, I mean, people did surprisingly well. And, for example, if you look at head motion, which is a big confound in fMRI, people actually moved less than in previous fMRI studies, which was probably because of the fact that they were, fMRI psychedelic studies,
Yeah.
because of the fact that they were familiar with the environment.
Hmm.
Um, but I'll tell you a couple of the results that I think were the most interesting from the acute drug imaging sessions.
Mm-hmm.
Um, One was, we replicated the observation and showed pretty compellingly that, there is this big change in the default mode network, which, like I mentioned, there was this was not the first time this idea was put forward, but we showed that in a pretty compelling way that across individuals, that seems to be the area that's the most disrupted by a psychedelic. Um, and in fact, there is a sort of common change in connectivity that's pretty consistent across people that actually very strongly correlated with some of these uh, subjective effects, like the loss of ego, for example, and the feelings of, connectedness and the disruption in the sense of in the sense of space and time and self, those people's subjective report of those sorts of changes was very strongly correlated with the degree to which default mode network connectivity was altered. So that's one thing. But actually, the thing that that I like the most, and that's why we titled the paper "Psilocybin Desynchronizes the Human Brain," is that I think that's like maybe a first level observation is that connectivity has changed. But the second level question is, well, how is connectivity changed? What's actually going on at the level of neural activity? And when you look at neural activity, and you remember a little earlier I mentioned that this fundamental principle that you have uh, brain areas that are highly synchronized and organized under normal conditions. And what's actually going on is that when you give a high dose of a psychedelic, these brain areas, at least the parts of the brain that are particularly high in the serotonin 2A receptor, they become desynchronized. Okay. And so, let's say you take 100,000 neurons, makes up a brain area, and under normal conditions, at any given time, 90% of those neurons will be firing in very close synchrony to each other. And under a psychedelic, it just becomes totally desynchronized, and all of these neurons are basically out of sync with their local field and with their neighbors. And that, I think part of the reason I think that that's a interesting and important observation is because, for one thing, it fits very well with what's been observed, at across very different experimental methods. So there's actually, I mean, I'm talking about results from an fMRI study, but there are actually studies recording from single units and from the local field, and showing that a psychedelic does this exact thing of desynchronizing single units with their local field. Um, so I think it's compelling for that reason, but I also think it probably has very important a relationship to both, what is the sort of nature of the psychedelic experience, but also to why it is that psychedelics produce this lasting plasticity in these particular parts of the brain that are related to self and related to emotion.
Hmm. So one of the convenient side effects of your experimental design here, as you said, was people had to go through many sessions in the scanner. So you had more data and you can make use of it in different ways, but also they got used to the scanner. And so they actually moved around less. And a challenge with fMRI is, right, the more someone moves their head around, the more difficult it is to actually use and work with the data. So a relative lack of head movements is great because you've got less of a concern about the artifactual fMRI signals you might get from head movements. One thing I have to ask before we go to more results, just because I am a former systems neuroscientist myself, is how do you track or deal with eye movements that might differ systematically between the psilocybin and non-psilocybin condition here?
Well, when people are in the scanner at rest, we basically just have them staring at a little crosshair in the middle of the screen, and we are recording their eyes both to make sure they're awake and to make sure they're okay, and staring at the crosshair. So that we are relatively controlling for.
I see. So they're instructed to maintain their gaze on this fixation point, and you are actually recording. You're actually able to record to video their eyes so that you have some sense of how much movement was there and when.
Mm-hmm. Mm-hmm.
Interesting. So, so you replicated the basic result that psilocybin leads to this desynchronization and a particular type of effect on the default mode. The default mode network generally gets more quiet, and that effect more or less scales linearly with the subjective report people give for how intense the trip was.
Yeah, I mean, I wouldn't use the term more quiet because that implies that the default mode network is deactivated.
Mm.
It's not deactivated really, it's just disorganized, and it's not activating in a synchronized way like it normally is.
Mm-hmm. And So this desynchronization effect, how does that compare to other drugs? I believe the control condition you guys use here is methylphenidate. How does that comparison look?
Well, methylphenidate didn't cause desynchronization at all. Um, that I can tell you with confidence. You know,
And this is ADHD medication, right? Ritalin?
yeah, methylphenidate, yeah, right. So it's Ritalin. It's generic Ritalin. The reason we chose that for uh, our placebo is because we wanted to match for arousal effects of a psychedelic. So, psychedelics inherently increase arousal, they increase blood pressure. And so we kind of matched the dose of methylphenidate to cause same effects in blood pressure. But it certainly did not, I mean, it didn't cause desynchronization at all. If anything, maybe slightly increased synchronization.
Hmm. And so, other than replicating sort of the basic results that had been seen before around this desynchronization effect, did you notice anything new in terms of how connectivity patterns changed, or different parts of the brain that you were able to pick up that other studies based on their design couldn't pick up, maybe because the effects were too subtle?
Well, yeah, I mean, there's one thing that came out of this that I really liked was that in terms of the lasting effect of the drug in the weeks after it had worn off, you see this relatively specific change in connectivity between the default mode network and the anterior hippocampus. And so this is a circuit that is associated with so hippocampus is memory, default mode network is sense of self, and we think of the circuit between them as self-referential memory, or the link between them as related to self. And there's some interesting studies showing that this particular connection is related to self-referential memory. And uh, in the weeks after psilocybin, this connection was decreased. So you have a reset of this kind of self-referential memory circuit um, pretty consistently across participants. So, I mean, that's one, I don't know, it's the first thing that comes to mind, but it's one example of, by actually mapping out these systems pretty specifically in individuals, we were able to see that there's really this reliable effect.
Mm-hmm. And I think one of the things you did here that either hasn't been done before or hasn't been done much before is you weren't simply passively imaging people on and off psilocybin. There was also a task engagement portion to this study. So when people are on psilocybin, how does the brain look? What are the changes that you see when they're engaged in a task compared to when they are not engaged in a task?
Yeah, this was another cool observation. So we had people, okay, so let me set up the design a little bit first because we had people get in the scanner, like I was saying, they're staring at a little plus, but basically they're letting their mind wander and having whatever psychedelic experience they're going to have. And then after maybe 15 or 20 minutes, we would say, okay, we want you to do this task. And this is a task they had already, practiced kind of when they were in the scanner during the baseline scans. But essentially it's a pretty easy visual matching task. So you see a picture, you hear a word, and then you make a judgment if the picture and the word match. Okay, so it's easy enough that you can do it when you're on a hero dose of psilocybin, um, but it requires you to be paying attention to your present environment. And what we saw was that when people are, when they switch from mind wandering to suddenly focusing on this task, actually the effects of the drug dramatically decreased.
Mm.
Okay, and I love this because there's this, idea of grounding, right? Which is this idea. I think I first heard about it in the context of dealing with overpowering emotions. That by bringing your attention to your kind of perceptual surroundings, you can take the brain out of this, out of whatever internal state it's in and re-normalize, brain activity, re-normalize emotional state. And this idea of grounding has also been certainly invoked with psychedelic therapy, that if somebody is, what tends to happen in a very intense psychedelic experience, maybe both positive but also negative, is that you get this like loop, right? Where you might feel, I guess, a bad trip is probably the easiest way to for me at least to conceptualize is you start having this thought like you're going to die. Next thing you know, your body feels like you're physically dying, and there's like seems to be this spiral where things get more and more intense when you're just in in a psychedelic state, right? But by forcing someone's attention to their perceptual environment, you can actually clamp down the intensity of the psychedelic experience. So we saw a very nice parallel to this where when people, were forced to engage in this pretty easy task, um, these observations, so the desynchronization, the change in default mode network connectivity, they actually got a lot smaller. So there's this nice sort of task by drug interaction.
Mm. And this fits with what I think a lot of people who have personal experience with psilocybin or other psychedelics often find. If you close your eyes while you're sitting
Yeah.
or lying down, the trip will often become harder or at least more intense, not necessarily harder. But then if you engage with the external environment, you touch things, you focus on music or whatever, this brings you out of the deepest parts of the
Yeah.
trip usually.
And there's a cool study. I'll have to think about who the first author is, but I know it's from, Robin Carhart Harris's the lead author, where they interrupt people every minute. I think it was DMT. We have to look it up. But they give a short acting psychedelic and they interrupt people every minute throughout the experience to ask them to report drug effects and to ask them to, answer questions. And then at the end, they ask them, how intense was the experience? And in the group where they interrupted people, uh, it's a much less intense experience than in the control group. They just, wait till the very end, right? They don't interrupt them and they let them have their psychedelic experience. And then at the end, they ask them about the experience. And it's less intense. It's less, people use the mystical experience questionnaire, which has been a pretty good correlate of therapeutic benefit. It's less of a mystical experience. So there is, there's this nice, like you're describing, kind of this idea of grounding that fits very well with what we saw in the brain.
Mm-hmm, mm-hmm. And so when you do these studies in humans, obviously we're talking about the human brain, the human experience on psychedelics, where there's a whole other world of the animal models and using psychedelics and animal models to understand things on a more mechanistic level. How this is a little bit of a vague question, but I just want to probe it a little bit. So obviously humans and animals are different, and yet we share many commonalities. So, you give a laboratory rat or mouse a psychedelic like psilocybin, and they have some kind of a trip, right? They have a behavioral response you can measure. They have changes in their brain that you can measure. They have inductions of plasticity that you can measure. They have 5-HT2A receptors and roughly the same pattern of expression that we do. But of course, none of that is identical. And in fact, one of the things that I think is interesting is that among other things, one of the big differences between the human brain and the non-human and non-primate brain is that although they have 5-HT2A receptors, although they respond to psychedelics in a directionally similar manner, some of the biggest differences between, say, a rodent brain and a human brain involve areas of cortex that are greatly enlarged in humans relative to rodents and other animals. And some of these have some of the highest density of 5-HT2A receptor expression. So I'm just wondering if you could comment a little bit about sort of how we should think about the extent to which the animal results translate to humans and vice versa, and where we should be particularly cautious.
Yeah, no, I think it's a huge issue. And I think that, I agree with everything you said. The ability that we can to say anything intelligent from what's a psychedelic doing in a mouse is limited. And, I mean, I think a lot of the things we're talking about with the subjective psychedelic experience are very difficult. I mean, first of all, they're difficult to measure in a mouse, much less to even know that, there is even a proxy or a parallel to the effect in a mouse. That's totally open questions. And I think, one of the sort of reasons that it's imperative to be able to do some of these studies in humans. So, I think the plasticity idea has been a big influence in psychiatry, and this is just this broader idea that you know inducing plasticity is probably critically relevant to the treatment of depression and the treatment of maladaptive psychological states, and that I think we there's enough evidence now to really believe that that's not just the case in mice. It's like it's a fundamental truth across models and across species. But beyond that, that still leaves a huge question about, we've seen that when you give a mouse a psychedelic, you have this activation of what's called the neurotrophic cascade, which includes things like brain-derived neurotrophic factor, and all these other markers that have been, measured and understood, um, and kind of ends with the formation of new synapses, which has also been measured in stunning detail in in live rodents. But it's still like totally an open question of what does that mean in a human? What does it mean to have neurotrophic cascade activation or to have plasticity? And ultimately, it's a those are questions that we're only going to answer with humans, uh, just because there has to be a psychological consequence to these big changes, but it's not going to be possible to say what it is just from rodents.
Mhm. Yeah, and it's one of the challenges of trying to look at and compare and translate the human stuff from the rodent stuff is, and the frustrating things is that we've got thousands and thousands of studies in rodents mainly where sure they have what we call depression like behaviors and anxiety like behaviors, and you can give them drugs, you can give them antidepressants, you can give them psychedelics, and you can measure what seem to be therapeutic behavioral effects and all sorts of exciting things. And yet, if I'm going to caricature things a little bit here, maybe, but if you just look at the past few decades of research in psychiatry, trying to translate from rodent work to human work and trying to come up with new drugs that do better at treating things like major depression and other psychiatric ailments in humans, we've got thousands of studies with all kinds of stuff that look promising, and then they just don't translate to humans. And one wonders to the extent to which our animal models are truly modeling something like major depression, where someone is ruminating and thinking linguistically, and there's a lot there that the mouse brain just probably doesn't do.
Yeah, completely agree. I think one of you know, this whole neurotrophic hypothesis, just to come back to that for a second, like one of the big goals, and to me, like a holy grail of sort of modern psychiatry and psychiatry theories, is to be able to measure plasticity in humans, and to actually, and which we've done, we've achieved it 100 times in mice, but to actually measure it in the human brain, uh, for exactly that reason. Because if we know, and know is probably a strong word, but we can assert that drugs that or treatments that make the sort of this core machinery that's related to depression more adaptable and more plastic are going to be therapeutic. And like you're saying, we've now found 1,000 drugs that do this in mice, but it's much harder to determine if those drugs are going to have that effect in humans. And if we could give a drug to, let's say, 4 people and say with confidence, yes, this drug is taking these circuits that are related to mood, and just to back up here, I mean, like, we have this idea of the cognitive behavioral model, which is the idea that you have emotion, behavior, and thoughts or cognition are in this kind of self-perpetuating loop. And when you're in depression, this self-perpetuating loop is in a very negative state, right? And the consequence is you don't want to get out of bed. You have, a pessimistic view of the world, and you feel lousy, right? And you seem to be stuck in that state. And so, I think it's pretty well understood now that one of the core pieces of treating depression is putting the brain into a more adaptable state so that it can get out of those thoughts, right? Those maladaptive thoughts. Um, and one of the, I think like holy grails of modern neuroscience related to psychiatry is to actually be able to measure the degree to which a drug puts the brain into a more plastic and adaptable state. And there's this great, and we don't have a tool to do that yet, which is actually surprising because it's been harder than you might have thought. Because, like I said, there's so many ways to do it in mice. But you would think there would be an easy, let's say, PET imaging biomarker that would say, here's how much BDNF is in your brain, or here's
Mm-hmm.
how quickly your brain is forming new synapses. But it's been pretty elusive. Um, but there is cool studies, for example, in pigs. So at least pigs are probably a lot closer to humans than mice. And you give a pig a high dose of psilocybin, and you actually get like a 10% increase in synaptic density
Hmm.
in the hippocampus, which is wild to me.
That's a
Um,
pretty big effect, I think.
yeah, yeah. And something in the order of, 6% increase in synaptic density in the medial prefrontal cortex. Um, so that's getting at least a little closer. But this is all still like, post-mortem studies, so not exactly techniques that could easily be scaled up to humans.
Hmm. And remind me, so you have training in psychiatry as well, right?
Yeah, so I've MD, PhD, and did residency in psychiatry.
Hmm. So as someone with that sort of dual background in functional imaging, human systems neuroscience, and psychiatry, when you think about taking this stuff from the laboratory to the clinic, when you think about the nature, the inherently subjective and powerfully subjective nature of these drugs, and you think about things like the procedural requirement to have placebo control groups when you're trying to do clinical trials to get things approved by the FDA. What do you think about? So maybe we'll just take the MAPS studies that the FDA didn't approve recently. I believe one of the key things there for the FDA was that you didn't have what they considered to be a true placebo control group. Given the nature of these things with when it comes to subjectivity and the subjective effects and your psychiatric background, how do you sort of think about doing placebo control studies, or whether we need to rethink that perhaps when it comes to thinking about whether to take these things to the clinic and start using them on a larger scale?
Yeah, that's been a big issue. And I can't even decide how to respond because I totally appreciate both sides of the argument. So let me like build both sides very quickly of the
Mm-hmm.
argument, and then we can pick which side we like. So on the one side, and I really like depends what day you ask me. I believe both things. On the one side, it's like a ridiculous idea that I should be able to control for this powerful transformational experience, because what does that even mean? Like it is fundamentally the combination of expecting that you're going to have a powerful transformational experience, plus having a very salient, very powerful experience uh, that enables healing and enables somebody to, I think both of those are like important parts of recovering from a psychedelic treatment or with a psychedelic treatment. Um, and to label like the to label that a placebo effect, it's almost like, well, okay, fine if you want to, but it works, right?
Mm-hmm.
So who cares if you label it a placebo effect?
Yeah, and I think
Does that make sense?
one thing that's worth pointing out here, at least for me, is what we might call an orthodox sort of clinical view would be that, well, there if we're talking about the brain and say curing someone's depression or something like that is going to involve triggering a certain pattern of brain activity that makes them better. I'm just going to call the orthodox clinical view might be something like, well, we need to have a placebo control group to show that it's not merely or largely someone's expectation or desire to get better that is actually making them better. But what's problematic about that is the effect of the drug coupled with things like your desires and your expectations will actually induce a different pattern of brain activity, which may, so someone who wants to get better and expects to from the drug is going to have that expectation coupled to the drug effect. That's going to induce one pattern of activity. And if I'm highly skeptical or I'm just, rigid and I don't necessarily have the motivation to get better that the other person does, that's going to mix together with the drug effect and give a different overall pattern of brain activity, which itself, I mean, if we're talking about patterns of brain activity, that is sort of the medicine here in some sense.
Yeah. And there's I'm blanking on his name. He's an anesthesiologist at Stanford. I can't remember.
Boris Heifetz.
Thank you, Heifetz. Yeah, he uses this great example of this in, I heard him give a talk at ACNP conference last year, and he described this study that's, I think it was like from the 1940s, 50s maybe, where they just give epinephrine, and then in one group they, basically prime them to believe it's going to be a very positive, joyful experience, and in the other group they prime them to believe it's going to be a very negative and sympathetically activating, angering experience, and you get those exact effects. So they use, it's kind of a fun design because they use what do you call it? They put someone in the room who is a Confederate who's in on the experiment, right? And have them react in different ways to the drug. And but the point that he's making is that, is just what you're saying, which is that the expectation of the effect of the treatment can dramatically influence the effect of the treatment. And then there's another side to that with psychedelics that is an equally big issue, I think, which is that if let's say I'm recruiting for my psychedelic clinical trial, well, who's coming to, seek out my trial? Well, some portion of them is going to be people who have heard that psychedelics are a great treatment for depression, or who have had an experience with psychedelics before that led them to believe personally that it might be a promising treatment. And then you take those people, and now half of them are randomized to receive a placebo. And so you have also this kind of effect where they are going to be disappointed and, have a worse reaction than they might because of the expectation that they were going to benefit from a psychedelic is actually causing them to do even worse on a placebo than they would if you had never given them the expectation that they were going to get a psychedelic in the first place. So the fact that we're so bad at blinding in psychedelic clinical trials is like a double hit because of the way that it's affecting the arm that gets the psychedelic, and because of the way that it's affecting the arm that gets the placebo.
Right.
So that's the other side of the
yeah. So it can actually sort of exaggerate the difference between the two groups.
Yeah,
on both sides. Interesting. Yeah,
but it's like, so like, I mean, just to bring it back to this point of maybe we shouldn't be so obsessed with this, is there's nothing wrong with, I mean, one of the fundamental principles of psychotherapy is setting the expectation that this treatment is going to help you get better. So you have to use the placebo effect, and it's effective. It's huge in psychiatry. And so why would you not use it, right? Why would you not set the expectancy that a treatment is going to help you?
Yeah. But, and again, from my perspective, setting that expectation, it's not, as you're saying, it can be used as a tool intentionally, and the setting of that expectation is going to change the functional brain context in which the drug acts, which will result in a different pattern of functional stuff happening in the brain than taking the drug in the absence of that expectation. And I think it's entirely reasonable and plausible to suppose that, to a large extent, the dual combination of a drug and an expectation might be necessary for therapeutic value to actually emerge.
Totally. And there's some good examples of this too, with, if you know the history of psychedelics, with like
the full version of this episode is available to paid subscribers at the Mind and Matter Substack at mindandmatter.substack.com. The full version is also freely available on my YouTube page. Just look up Mind and Matter or my name, Nick Jikomis, on YouTube, and you'll find the episode there. This is a partial version of the episode, and so it'll cut off partway through, but you'll still get a significant portion. So sign up to become a paid subscriber at Substack or visit my YouTube page to get the full episode. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.